Hello everyone, this is a topic about cell cycle. Let's go to the study of section 1. What is cell cycle? It is known that a sperm of a human being looks like a tadpole, which is very small, and the egg is not big either. A zygote is the first cell of a new person, which can form a baby much bigger than a cell. In this process, cell division cycle is necessary. The cell cycle is a process in which a cell grows and divides to create a copy of itself. It is a process from the end of cell division to the start of the next cell division. Cell cycle entails an ordered series of macromolecular events that lead to cell division and the production of two daughter cells, each containing chromosomes identical to those of the parent cell. In animals, the cycle takes around 24 hours from start to finish for most cell types, although some, such as those in the skin, are constantly going through this cycle, while others may divide rarely, if at all. Neurons, for example, don't grow and divide once they are mature. Abnormal cell cycle is basis for cancer studies. The primary events of a mammalian cell cycle are replication of chromosomes and their segregation to daughter cells, extraordinarily high fidelity. According to this, cell cycle can be divided into interphase, which includes G1 S, G2 phase, and division, which is equal to M phase. M phase includes mitosis and cytokinesis. There is G0 phase for cells not divided, which perform particular functions and can return to the cell cycle when properly stimulated. The G1 phase is set in immediately after the cell division. It is characterized by a change in the chromosome from the condensed mitotic state to the more extended interphase state and by a series of metabolic events leading to initiation of DNA replication. During G1 phase, the chromatin fibers become slender, less coiled, and fully extended, and more active for transcription. The transcription activities result in the synthesis of ribosomal RNA, transfer RNA, messenger RNA, and a series of molecules required for the initiation of DNA synthesis. The length of this phase is more than the other three phases. After G1 phase, there comes the S phase. It is the phase of active DNA and histone synthesis. During S period, doubling of the slender fully extended chromosomes takes place, which is accomplished by the doubling of DNA and the associated proteins in the chromosomes. The replication of DNA is semi-conservative and discontinuous. Although some of the histone protein is synthesized during G1 phase, most of it is synthesized during S phase. Further, synthesis of ribosomal RNA must continue from G1 to S phase if DNA synthesis is to start. G2 phase follows the S phase. The critical event is concerned with the condensation of chromatin. The synthesis of RNAs and proteins occur during this process. Mitosis is relatively brief and typically takes only about 1 hour. This is the process by which the two identical sets of DNA are physically separated from one another, forming two sets of chromosomes, which are then drawn to different ends of the cell to form separate nuclei. During this phase, the originals, such as mitochondria, also divide. The final stage is cytokinesis. This is when division into two distinct units actually takes place. Opposite cell walls are drawn toward one another at around the middle point until they meet, forming two units that split off from one another. In M phase, mitosis stage are composed of prophase. The chromosomes appear condensed and the nuclear envelope is not a parent. Metaphase, 6 coiled chromosomes, each with 2 chromatids, are lined up on the metaphase plate. Anaphase, the chromatids of each chromosome have separated and are moving toward the poles. Telophase, the chromosomes are at the poles and are becoming more diffuse. The nuclear envelope is reforming. The cytoplasm may be dividing. So, the cells in vivo can be divided into 3 categories. Cycling cells that are dividing continuously, for example, stem cells. G0 cells that don't divide normally, but divide when given an appropriate stimulus, for example, liver cells and lymphocytes. Terminally differentiated cells, which are highly specialized and have lost the ability to divide until they die. For example, muscle cells, red blood cells, and nerve cells. A multiple cellular organism needs to coordinate cell division across different tissues and organs, which is critical for normal growth, development, and maintenance. Even the cells within an organism's body divide and develop at different rates. Cell cycle events are triggered by the cell cycle control system, which is a set of molecules found in the cytoplasm affected by inter- and external controls. External factors include the presence of essential nutrients, growth factors, which are proteins secreted by certain cells that stimulate division of other cells, density-dependent inhibition, in which crowded cells stop dividing since chemical requirements are insufficient to allow cell growth, but cancer cells lose it. Authority dependence, that is the need for cells to be in contact with the substrate in order to replicate. DNA damage can inhibit cell growth. An experiment can demonstrate that cells fail to divide if an essential nutrient is left out of the culture medium, for example, growth factors. Another experiment can determine authority and density-dependent inhibition in animal cancers grown in culture. To divide, the cell must be attached to a solid surface, and crowded cells stop dividing. Proliferating cells in culture tend to double in mass before each division, but cell size at division is variable and depends on the concentrations of extracellular signal proteins that stimulate cell cycle progression, cell growth, or both. So, cell growth determines cell size, while cell division and cell death decide cell number, which in turn together suggests the total cell mass and thus organ size or body size. This is the end of the section. Thank you very much.